So I want you to imagine that your stomach lining is this bustling, highly organized city.
Right, like a microscopic metropolis.
Exactly. And every single cell in this city has a very specific job, a recognizable uniform, and a strict set of daily instructions.
It's a harsh environment, though.
Oh, totally. I mean, it is full of acid, churning food, but the city functions perfectly under normal conditions.
Until it doesn't.
Right. Now imagine a scenario where chronic stress and relentless inflammation just repeatedly hit that city. The environment goes from harsh to completely toxic.
And the cells have to react.
Yeah, to survive, the city's construction workers do something drastically unexpected. They panic.
They realize the old ways aren't working anymore.
Exactly. They essentially throw away the old blueprints and start building a completely different type of city right on top of the old one. They swap their uniforms, they change their tools, and they start building intestinal structures where, stomach structure should be.
It's a desperate defense mechanism.
Yeah, it really is. But here is the terrifying part. This organized survival strategy eventually spirals out of control, building the chaotic, destructive landscape that we know as cancer.
It's such a striking way to visualize what is fundamentally a microscopic identity crisis.
An identity crisis, yeah. I like that.
Because we are talking about deep cellular survival mechanisms that start out as protective, right? But eventually they go horribly, horribly wrong.
And that identity crisis is exactly what we are unraveling today. Welcome to the Deep Dive. We are looking at a really groundbreaking 2026 study published in Nature's NPJ Precision Oncology Journal.
It's a dense paper, but the implications are massive.
Absolutely. The researchers essentially caught stomach cancer in the act, literally cell by cell.
Which is incredibly rare to see with such clarity.
Yeah, so our mission for you on this deep dive is to trace the exact step-by-step origin of intestinal-type gastric cancer. We're going to map out the biological mechanisms, the cellular shifts, and the specific genetic switches that get flipped as a normal stomach cell slowly goes rogue.
And what makes this study so powerful is the technology they use to do that mapping.
Right, the single-cell sequencing.
Exactly. For a long time, we knew the general stages of how stomach cancer developed, but we couldn't really see the specific molecular foreman directing the construction site, to use your analogy.
We were just guessing from the outside.
Yeah, but now, using single-cell RNA sequencing, we can see everything.
I always think of single-cell RNA sequencing like this. If traditional genetic sequencing is like tasting a fully blended fruit smoothie, to guess what ingredients went into it.
Which is pretty hard to do accurately.
Right, you get a general sense of it. But single-cell sequencing is like a magic machine that separates that smoothie back out into individual strawberries, individual blueberries, and bananas.
That's a great way to put it.
You aren't just tasting the whole mix anymore. You can examine the exact genetic recipe that is active inside every single piece of fruit.
And that exact level of granularity is what allows us to track a very well-documented sequence of events known as Correa's cascade.
Correa's cascade, yeah.
It's basically the biological pathway from a normal stomach lining all the way to gastric cancer. But, to understand how the cells go rogue, we first have to understand their normal baseline.
I actually use a little memory trick to remember the stages of Correa's cascade.
Oh, really? What is it?
It's the phrase, never call Iggy early.
Never call Iggy early.
Yeah, NCIE. That gives you the 4 major phases we're going to talk about today. Non-atrophic gastritis, chronic atrophic gastritis, intestinal metaplasia, and finally, early gastric cancer.
Okay, I like that. So let's start right at the beginning with the non-atrophic gastritis, or NAG.
Our baseline.
Exactly. This is our baseline, and we're looking specifically at the lower part of the stomach, which is called the antrum.
Down at the bottom there?
Yeah. And down there, you have what are called progenitor cells, or PCs.
Okay.
Think of PCs as undecided college students. They hold a ton of potential, but they haven't picked a major yet.
They're just taking general ed classes.
Pretty much. Under normal conditions, they generally differentiate or pick a major into one of 2 mature cell types. They either become pit mucous cells or gland mucous cells.
And using that single-cell sequencing, the smoothie separator, the researchers essentially built a family tree of these cells over time to see what majors they were picking, right?
Using a tool called Monocle 2, yes, it maps out pseudotime.
Right. And it turns out at this baseline NAG stage, most of those undecided progenitor cells choose to become pit mucus cells.
The PMCs, yeah.
But why? Like,
Yeah.
what is actually physically happening inside the cell to make that specific choice?
Well, it all comes down to transcription factors.
The directors.
Exactly. These are specialized proteins that literally enter the cell's nucleus, bind to the DNA, and turn specific genes on or off.
So they're like the academic advisors handing the student the syllabus and saying, you are taking these classes.
That's spot on. In this baseline stage, a transcription factor called TGLN2 takes the lead.
TGLN2.
Yes. And it works alongside another protein called AGR2 to push the progenitor cell down the path to becoming a pit mucus cell. It is a highly controlled, very stable environment.
But of course, biological environments rarely stay stable forever.
Unfortunately, no.
Which brings us to the C in our cascade.
Yeah.
Chronic atrophic gastritis.
CAG.
Right.
Let's say the stomach lining is facing severe ongoing inflammation, maybe from a bacterial infection, like H. pylori, or just extreme dietary stress over a long period.
The native glands of the stomach start taking actual physical damage.
They shrink and atrophy, right?
Yes. And the cellular workforce realizes that the old blueprints are failing. The baseline programming simply isn't keeping them alive anymore.
The environment changed.
Dramatically. So the progenitor cells pivot. Instead of turning into pit mucus cells like they used to, they drastically shift their output to favor gland mucus cells.
So they are actively changing the factory output just to survive the new conditions.
Yes, and that means the old academic advisor, TGLN2, isn't working anymore.
They need new directors. Exactly. The sequencing data shows that two new transcription factors step in here, SBZEF and KLF10.
SBDEF and KLF10.
Right. They essentially silence the old pathway and force the undecided cells to aggressively become gland mucus cells instead. It's a direct adaptive response to the physical destruction of the stomach lining.
Okay, before things get even more chaotic in the city, I want to do a quick recap, make sure we're all holding these pieces together.
Good idea.
So we've got our baseline, the NAG stage, where progenitor cells are just trying to be pit mucus cells guided by KGLN2.
Correct.
But when chronic inflammation causes the stomach tissue to shrink and atrophy the CAG stage, those cells hit a panic button. They swap their genetic directors to SBDEF and KLF10, and they start pumping out gland mucus cells instead just to survive the damage.
That is the crucial takeaway so far. The cells are adapting.
Which leaves you wondering, if your stomach cells are already rewiring their default programming just to survive an inflammatory attack, what happens when that attack never stops?
Well, the environment eventually becomes so hostile that being a stomach cell at all is a liability. And that pushes us right into the I of our cascade, intestinal metaplasia, or IM.
This is where things get really weird.
Very. The stomach cells essentially decide to put on heavy intestinal armor.
Intestinal armor?
Yeah, they attempt to transform into intestinal cells, specifically absorptive cells known as enterocytes and secretory cells called goblet cells.
I always used to think this was just, a bizarre mutation, like a stomach cell gets blasted with inflammation and suddenly just mutates sideways into an intestine cell.
A lot of people think that, yeah.
But looking at the sequencing data, it's not a random sideways jump at all, is it?
Not at all. They actually have to move backwards first.
Backwards.
Right. I mean, biology is usually a one-way street, Cells mature, they specialize, and that's it. But here, the single-cell data revealed a massive identity regression.
They de-differentiate.
Exactly. Both those undecided progenitor cells and those newly formed gland mucus cells undergo de-differentiation. They regress into a blank slate stem cell-like state called intestinal-like stem cells, or ILSCs.
So it's literally like taking off a highly specialized uniform. But physically, how do they do that?
The transcription factors have to essentially fold up the sections of DNA that contain the stomach instructions and lock them away.
They just hide the manual.
Yeah. And by silencing those stomach genes, they unearth an older, more primitive genetic state. We can actually track this happening in the data because these transitional blank slate cells light up with a very specific gene marker called OLFM4.
Okay, so picture this stomach cell stripping off its work uniform and putting on a blank gray T-shirt.
A gray T-shirt.
Yeah, that gray T-shirt is the OLFM4 stem cell phase. It has to go back to basics wearing this plain shirt before it can start putting on the heavy defensive armor of an intestinal enterocyte.
That's a perfect visual, and putting on that new armor is an incredibly energy-intensive process.
You bet.
The study actually maps exactly how these gray T-shirt cells mature into enterocytes through two distinct pathways.
Okay, let's break those down.
For the progenitor cells, the transformation is driven by a transcription factor named HNF4A.
HNF4A, what does that one do?
What HNF4A does is execute a total metabolic reprogramming.
Metabolic reprogramming. So it's not just changing what the cell looks like, it's changing what it eats.
Precisely. HNF4A fundamentally alters how the cell processes lipids and how it generates energy. It changes the cell's internal diet so it can function like an intestinal cell.
That is wild.
Meanwhile, the gland mucous cells take a slightly different pathway to get to the same destination, driven by a factor called ESRA.
ESRA, does it do the same thing as HNF4A?
It's related, but different. ESRA interacts with a network of cofactors, specifically the PGC1 family, to heavily regulate cellular energy production.
Because building armor is exhausting.
Exactly. A cell undergoing this kind of drastic identity shift requires a massive amount of fuel. ESRA ensures the cell has the energy reserves to completely abandon its old stomach identity and sustain the new intestinal one.
And while all of this is happening, there is a whole parallel assembly line running in the background, right?
But for the goblet cells?
Yeah, because an intestine isn't just made of absorptive enterocytes. It needs secretory goblet cells to make mucus, too.
Right. And the data shows that the secretory cells in the stomach undergo their own regression and transformation, eventually becoming fully functioning goblet cells.
Driven by different directors, I assume.
Yes, a different network of genetic switches like CRF3L1 and AFOXQ1, but the overarching theme is identical.
It's a coordinated, desperate attempt to build a completely different organ just to survive the toxic environment.
It is a truly remarkable display of biological plasticity.
Okay, time for another quick 5-minute recap, synthesizing this middle leg of the journey.
Go for it.
So the inflammation's gotten so bad that the stomach cells just throw in the towel. They regress backwards into a stem cell-like gray T-shirt phase, the OLFM4 cells.
Right.
And then, guided by factors like HNF4A and ESRI, they completely rewire their own metabolisms, literally changing how they process energy and fat just so they can wear intestinal armor.
Spot on.
But this comes with a terrifying risk. I mean, if you have millions of cells that are so confused about their identity that they're unzipping old DNA and rewriting their own energy engines,
Yeah.
you have to ask, how close are they to losing the rulebook entirely?
They are standing right on the edge of the cliff.
Wow.
Because the intestinal armor strategy is just a temporary fix. The inflammation hasn't stopped.
The environment is still toxic.
Yes, so the DNA is continually accumulating damage. The cellular identity is totally fractured. And that brings us to the final stage, the E in your mnemonic, early gastric cancer.
EGC. This is the critical tipping point of the entire paper.
It really is the main event.
The researchers wanted to find the exact origin point of the cancer. Like, which specific cell from that chaotic armored city finally goes completely rogue?
And to figure this out, they used an analytical tool called InferCNV.
InferCNV.
Yes. It looks for copy number variations.
What exactly is a copy number variation?
Well, as cells get heavily damaged and try to replicate, they make mistakes.
Typos in the blueprints.
Exactly. They might accidentally duplicate a massive section of their genetic instruction manual or delete a crucial page entirely. These large-scale structural mutations are copy number variations.
So using InferCNV is kind of like searching for a suspect by matching crime scene DNA to a database of known offenders.
That's a great analogy.
They compared the heavily mutated DNA of the known cancer cells to all the other struggling cells in the stomach lining.
And they found a match.
Who was it?
The cells that shared a staggering 0.966 correlation with the cancer cells, meaning they were practically identical, were the progenitor cells.
The undecided college students from the very beginning.
Exactly. But the single-cell resolution gives us an even sharper picture. We aren't just looking for any progenitor cell.
Right, because not all of them turn into cancer.
No. The researchers isolated the exact molecular mugshot of the cancer precursor. It is a very specific subset of progenitor cells marked by two heavily expressed genes, KAA0101 and PA1.
Okay, KO101101 and PA1. We need to unpack POAP1 because looking at the study, this seems to be the smoking gun.
It absolutely is.
Why is expressing PRP1 so dangerous for the cell?
Well, normal PRAP1 is a protein that just binds to triglycerides, but it plays a really sinister role in managing DNA damage.
Sister, how?
In a healthy body, if a cell suffers severe DNA damage that it absolutely cannot repair, a biological safeguard kicks in.
The self-destruct sequence.
Right. The cell undergoes apoptosis, which is programmed cell death. It basically sacrifices itself so it doesn't pass on corrupted DNA to the next generation of cells.
It hits the self-destruct button for the greater good of the city.
But PRAP1 physically blocks that button.
Oh, wow.
Yeah. When PRAP1 is overexpressed in these highly stressed progenitor cells, it prevents apoptosis. It gives the mutated cell a biological get-out-of-jail-free card.
So the cell should die because its DNA is completely corrupted.
But PRP1 keeps it alive, allowing it to bypass all the safety protocols and continue replicating.
It's literally ripping the wires out of the self-destruct mechanism.
Exactly. And once that ultimate safeguard is gone, new directors step in to run the newly rogue operation.
Who takes over now?
The paper highlights a transcription factor called SIRT6.
SIRT6.
Yes. SIRT6 is the engine of the malignancy. It is what we call an NAD-dependent enzyme.
Okay, let's pause there for a second, because whenever I hear NAD, I just think of those like cellular metabolism supplements that people take for anti-aging.
Right. NAD plus MET.
Yeah. What does it mean for SIRT6 to be NAD-dependent in the context of cancer?
Well, NAD is essentially a molecular currency for cellular energy. By being NAD-dependent, SRT6 burns through this energy currency to drive very aggressive cellular changes. It basically spends all the cell's money to do two critical things.
What's the first thing?
First, it pumps out inflammatory signals like TNF and IL-8, making the local environment even more chaotic.
And cancer loves chaos.
It thrives on it. Second, it alters the structural proteins of the cell, specifically increasing intracellular ADP ribose.
Which does what?
It gives the rogue cell the physical mobility it needs to migrate and invade other tissues.
Okay, so let me get this straight. PRIP1 keeps the heavily damaged cell alive when it should be dead, and SRT6 gives it the fuel and the mobility to spread out and attack.
It's an incredibly lethal combination.
And the researchers didn't just, look at this in a digital spreadsheet, right? They used something called spatial transcriptomics to prove it in physical space.
Yes. Spatial transcriptomics bridges the gap between molecular data and physical reality.
It's like a GPS.
Exactly. If single-cell sequencing tells us the exact genetic makeup of the rogue cell, spatial transcriptomics acts as the GPS system. It tells us exactly where that cell is living inside the physical tissue.
So they took actual physical slides of human stomach tissue and mapped it geographically.
They did.
And sure enough, those specific PRP1 and SIRT6 positive progenitor cells were found strictly co-localizing inside the physical boundaries of the malignant tumors.
They caught them right at the scene of the crime.
That is unbelievable.
It verifies everything. The genetic trajectory we tracked on a computer perfectly matches the physical pathology in the human body.
So to bring this whole journey together for our third recap, the origin of early gastric cancer isn't just some random lightning strike.
Not at all.
It traces back to a very specific subset of exhausted progenitor cells. And armed with POP1 to dodge cell death and fueled by SRT6 to drive inflammation and movement, these cells abandon their final safeguards and become early gastric cancer.
Which brings us to the clinical reality of all this.
Right. The so what? Why does this deeply granular molecular map matter to the average person listening to this?
How does knowing the molecular mugshot of these cells change your next trip to the gastroenterologist?
Exactly.
Well, it fundamentally shifts our approach from reactive to proactive.
Meaning what, practically?
Currently, we often have to wait to see a physical lesion or a tumor during an endoscopy before we know for sure that cancer has taken hold.
You're waiting for the building to be on fire before you notice it.
Right. But because we now have this incredibly detailed, stage-oriented framework, we know exactly what the precursors look like before the tumor forms.
We know the uniform they wear before they start building the tumor.
Exactly. In the future, specialized, minimally invasive molecular assays or endoscopic biopsies could be designed to screen specifically for the overexpression of PRA1 or SRT6.
So if you catch those rogue progenitor cells early enough,
you could intercept them. We could develop targeted drugs that block that ESRA energy pathway we talked about, or drugs that inhibit SRT6.
Essentially cutting the engine on the cancer's development before it ever crosses the finish line.
Exactly. It turns a potentially fatal diagnosis into a preventable, manageable event. It is a massive, massive leap forward for precision oncology.
It really is. As we wrap up this deep dive, I want to leave you with something to really mull over.
It's a lot to process.
It is. You know, we are conditioned to think of cancer as this chaotic, completely random breakdown of the human body. like a tragic biological
A
accident.
glitch in the system.
Right. But looking through the lens of this study, that isn't really true at all.
The precision of it is what's truly unsettling, yet totally fascinating.
Unsettling is the perfect word. It's an incredibly organized, desperate attempt by your own cells to adapt to a toxic environment. Step by step, transcription factor by transcription factor, they're just trying to survive by any means necessary.
They're doing what they think they have to do.
Yeah. And it really forces you to reconsider the permanence of your own biology. I mean, if cellular identity is this fluid, if a stomach cell can fold up its DNA, regress into a gray t-shirt stem cell, and try to become an intestine just to stay alive, are any of our cells ever truly fixed?
It's a profound question.
Or is every single cell in our body always just one severe stressor away from becoming something entirely different?
It reveals how astonishingly resilient life is at the microscopic level, even when that resilience ultimately becomes our own worst enemy.
That is so well said. Thank you for joining us on this deep dive into the hidden origins of disease. Keep asking questions, keep looking closer, and remind yourself to stay insanely curious.